16. Uptake and transport of mineral nutrients. Objective: to study about the uptake and transport of mineral nutrients. Uptake and transport of mineral nutrients. Plants obtain their carbon and most of their oxygen from CO2 in the atmosphere. However, their remaining nutritional requirements are obtained from minerals and water in the soil. Uptake of mineral ions. Unlike water, all minerals cannot be passively absorbed by the roots. Two factors account for this. They are: 1. Minerals are present in the soil as charged particles or ions which cannot move freely across cell membranes. 2. The concentration of minerals in the soil is usually lower than the concentration of minerals in the root. Therefore, most minerals must enter the root by active absorption into the cytoplasm of epidermal cells. This needs energy in the form of ATP. Respiratory inhibitors like cyanide inhibits ATP synthesis, generally reduce the ion uptake. The active uptake of ions is partly responsible for the water potential gradient in roots and therefore for the uptake of water by osmosis. Some ions also move into the epidermal cells passively. Ions are absorbed from the soil by both passive and active transport. Specific proteins in the membranes of root hair cells actively pump ions from the soil into the cytoplasm of the epidermal cells. Like all cells, the endodermal cells have many transport proteins embedded in their plasma membrane. They let some solutes cross the membrane, but not others. Transport proteins of endodermal cells are the control points where a plant adjusts the quantity and types of solutes that reach the xylem. Root endodermis, because of the layer of suberin, has the ability to readily transport ions in one direction only. Translocation of mineral ions. After the ions have reached xylem through active or passive uptake, or a combination of the two, their further transport up the stem to all parts of the plant is through the transport stream. The chief sinks for the mineral elements are the growing regions of the plant. They are apical meristems, lateral meristems, young leaves, developing flowers, fruits and seeds, and the storage organs. Unloading of mineral ions occurs at the fine vein endings through diffusion and active uptake by mesophyll cells. Mineral ions are frequently remobilized, particularly from older senescing parts. Older dying leaves export much of their mineral content to younger leaves. Early, before leaf fall in deciduous plants, minerals are removed to other parts. Elements most readily mobilized are phosphorus, nitrogen, sulfur, and potassium. Some elements that are structural components, like calcium, are not remobilized. An analysis of the xylem exudates shows that though some of the nitrogen travels as inorganic ions, much of it is carried in the organic form as amino acids and related compounds. Similarly, small amounts of P and S are carried as organic compounds. In addition, small amounts of exchange of materials does take place between xylem and phloem.